Okay, hope it's recording. The cell membrane is a highly mobile and dynamic structure made up of proteins, lipids, and carbohydrates. In high school, we may have learned two different concepts about the plasma membrane. One is that the membrane is very fixed or not fluid, and two, the lipids presented in the membrane make it very fluid. The first concept is wrong, according to our professor. A rigid membrane would not be able to function. Therefore, the membrane is actually very fluid. The membrane is also selectively permeable, meaning that the membrane controls what goes in and out of the cell, such as ions. Examples of ions would be sodium, potassium, chloride, and calcium. Then, of course, vitamins and nutrients. Some examples of nutrients would be carbohydrates and glucose. And then, of course, drugs and waste also have to pass by the cell membrane. So the cell membrane maintains a high amount of potassium inside, while there is a low concentration of potassium outside the cell. As for sodium, there is a small concentration on the inside of the cell, about 10 micromoles, while there is a high concentration of sodium on the outside, about 135 to 145 micromoles. The concentration of these two ions are maintained by the cell membrane, or more specifically, the transport proteins on the membrane, one of them including sodium, potassium, ATPase, but our professor also likes to call it NKA. Ion concentration in the blood and in the cell is very critical in maintaining normal physiology. An organ can die in minutes if this balance is disturbed. For our next topic, we're going to talk about the types of transport. The first one is going to be simple diffusion. Simple diffusion involves no membrane proteins. The molecules simply diffuse from high to low concentration. An example of this would be oxygen. When we breathe in, oxygen moves into the cell by simple diffusion. Facilitated diffusion is just like simple diffusion, except it involves a membrane protein to transport the substrate from high to low concentration. For example, glucose amino acids, these are not ions, however they are charged, meaning they will require a membrane protein to transfer them into the cell. And lastly, we have active transport. Active transport is the uphill movement of substrates from low concentration to high concentration, and this process does require energy, mostly in the form of ATP. So if we made a graph with the substrate concentration on the x-axis and the velocity as the y-axis, you would see that the solute transport will approach an asymptote. In other words, the solute is going to saturate at a certain level for facilitated and active transport only. The asymptote is going to be called Tm, or transport maximum, and then the Kt will be the amount of solute present at 1/2 of the transport maximum. For simple diffusion, the solute will never saturate, and this is because simple diffusion does not require a vehicle to transport solutes across the membrane, so particles will continue to move across the membrane basically forever. Transporters work by having the substrate bind to either the inside or the outside, which makes a conformational change to the transporter, which then either allows the substrate to move in or out of the cell. And then channels are protein tunnels that either open or close, allowing substrates to move in and out. This uniport involves one molecule and one protein. The molecule moves singularly in one direction. Next is the symport. This is a protein that allows two molecules to move through together in the same direction. then the antiport is a protein that allows two molecules to move through in opposite directions, driving one another. So sodium potassium ATPase pumps sodium out and then moves potassium in. So it would be an example of an antiport. But at the same time, it's also an example of active transport because it does need ATP to move ions against their gradients. So the book divided active transport into 4 different categories. The first one is F-ATPase. ATPase just means the hydrolysis of ATP into ADP, and this is always in the mitochondria and bacterial membranes. The next type is the V-type active transporters, and they are found in lysosomes. So we all know that lysosomes are vesicles that are highly acidic in nature inside. So to maintain this acidity, we're going to need V-type transporters to maintain the low pH inside the vesicles. Low pH basically means a high concentration of protons inside, and so basically the V-type transporters are going to maintain a high concentration of protons inside the vesicle, which in turn keeps the pH very low. The kidneys do a lot of excreting, and one of the excretions is the protons inside urine, which is why urine is acidic, and the V-type ATPase maintains this pH. Defects in the osteoclast, V-ATPase causes osteopetrosis, which is the increased bone density. The third type is the P-ATPase, which functions to maintain the cellular ion concentration. Nka is normally found on the plasma membrane and can drive molecules to be transported, such as sugars. So calcium ATPase can be found on the plasma membrane, but it can also be found on the sarcoplasmic reticulum right here. Calcium plays an important role in the contraction and relaxation of the heart, so a mutation in calcium ATPase will cause cardiomyopathy, and then NKA mutations will cause migraines. The fourth type is the ABC transporters, like P glycoprotein and MRP, function to excrete toxic metabolites and xenobiotics. So here we see calcium transport and mobilization in muscles. And the 2 types of muscles we're going to be talking about are the skeletal and the cardiac muscles. So as we can see, comparing the skeletal and the cardiac muscles side by side, we can see that calcium transport in the skeletal muscle is much simpler than calcium transport within the cardiac muscle cell. And this makes a lot of sense because the skeletal muscle cells just need to contract and relax, while the cardiac muscle must maintain a certain rhythm, which requires a lot of ion movement in and out of the cell. So when the skeletal muscle contracts, we're going to see depolarization within the cell membrane. This depolarization is going to activate the voltage-dependent calcium channel, or VDCC, as seen here. Once VDCC is activated, it will in turn activate the channels on the sarcoplasmic reticulum, or the SR. The cell normally does not have a high concentration of calcium inside because once calcium comes into the cell's physiology is changed. So the cell must store calcium in the SR. For calcium to leave the SR, it's going to go through the calcium channel through facilitated diffusion. And once again, that calcium channel is going to be activated by VDCC. So once the SR is activated, calcium is going to go through the calcium channel and it's going to then bind to calmodulin. And once it binds to calmodulin, calmodulin is going to be activated and it's going to activate the enzyme MLCK. And once MLCK is activated, it's going to then phosphorylate myosin. And after that, all of that happens, the muscle cell is going to contract. So after contraction, the muscle has to relax, meaning that calcium must go back into the SR. And for this to happen, we need calcium ATPase. Now moving on to the heart muscle, we're going to see depolarization as the first step as well, but unlike the skeletal muscle, depolarization of the cardiac muscle causes a little bit of calcium to enter the cell. So the calcium is going to move through the VDCC, and remember that the VDCC does not cause the movement of calcium into the cell. It's actually depolarization that causes the movement of calcium. So once calcium enters through the VDCC, it's going to trigger the release of calcium from the sarcoplasmic reticulum, the SR. And so once the calcium is released from the SR, it's going to in turn cause the cardiac muscle to contract. And once again, the calcium released from the SR must go back inside storage by calcium ATPase on the SR. Calcium can also leave the cell entirely by other transport proteins located on the plasma membrane. One of them, again, being calcium ATPase. And this can be found either on the SR or on the plasma membrane. The sodium-calcium antiporter, right here, also transports sodium in while pumping calcium out at the same time, and this requires no ATP. The sodium that enters through the sodium-calcium antiporter must then go out of the cell, because the concentration of sodium inside the cell must remain at around 10 micromoles. So the sodium leaves the cell through the sodium-potassium ATPase, or NKA, and this is going to require ATP. So our professor mentioned the calcium channel blockers, which are drugs that lower blood pressure by blocking the initial entry of calcium. This is going to reduce all of the events that I just mentioned, and it's going to cause the heart to beat less and work less. The next picture represents glucose transport in the intestine cells. The apical, or the intestinal lumen side, is going to have a lot of villi, and this is going to increase absorption of stuff. So on the membrane of the enterocyte, there is SGLT1. Do not confuse this with SGLT2. This is found exclusively in kidneys. So SGLT1 is going to transport glucose along with sodium inside the cell. Sodium must then leave the cell through, again, sodium-potassium ATPase, and then glucose is going to leave the cell through the GLUT2 membrane protein. Another monosaccharide, fructose, can also be transported into the cell through GLUT5, which is then promptly transported out through GLUT2, just like the glucose. So, what if sodium-potassium ATPase stops working? If it suddenly stops working, glucose would not be able to enter the cell because there would be no room for more sodium to enter the cell, carrying glucose along with it. And the very last picture shows acidification in the parietal cells of the stomach. So, protons right here, H plus, comes from the reaction of water and carbon dioxide. The potassium is going to be transported out of the cell through an antiport, and once the proton leaves the cell through this antiport, it's going to allow potassium to come into the cell. This potassium is then going to be transported out through a potassium channel seen right here. Now going back, bicarbonate seen right here is the second product of that reaction between water and carbon dioxide. So bicarbonate is going to be transported out of the cell, and in turn chloride is going to be pumped into the cell. So this chloride is going to move through a chloride channel seen right here out of the cell. Then the extracellular chloride seen right here is going to then meet with the proton that was initially pumped out right here, and they're going to combine to form HCl, or hydrochloric acid. And as we all know, hydrochloric acid is the acid that we're going to find in the stomach. So it looks like this is where he left off last time. Looks like I am done, and I'll see you guys next time.